name is Rupavi Devaluri, and today I'm going to be presenting my IRP on genomes. So these are my high-level questions. How do cells make RNA and proteins from DNA? How does the environment impact DNA and gene expression? And how can AI help find the meaning of a DNA sequence? So first, what is DNA? DNA stands for deoxyribonucleic acid. It is a sequence of 4 nucleotide bases: A, adenine, T, thymine, C, cytosine, and G, guanine. Nucleotide bases are like building blocks, but for DNA. DNA contains all the information needed to make any living organism. And adenine and thymine are always on the same rung. Also, DNA has this shape. It's called a double helix, and it looks like a twisted ladder. Adenine and thymine are always on the same rung of the ladder, and cytosine and guanine are always on the same rung. So you may be wondering, who discovered the structure of DNA? The structure of DNA was discovered by 2 famous scientists, Francis Crick and James Watson. James Watson used to live not too far away from here at Long Island at Cold Spring Hardwood Laboratory. My dad met him and has his autograph. The book with his autograph is I think it's on my desk. Okay. So I visited Cold Spring Harbor Lab, and James Watson worked as president of Cold Spring Harbor during those years here on Long Island, and I visited it here. I visited it in 2021, and here's a DNA double helix model. CF Seychelles has the DNA Learning Center, and I'll visit it. So, what is a genome? A genome is the entire set of DNA found in a cell. In humans, the genome consists of 23 pairs of chromosomes located in the cell's nucleus and another chromosome located in another part of the cell. A genome contains all the information needed for an individual to develop and function. So you may be wondering what a cell is, or what a nucleus is, or what a chromosome is. A cell is the smallest unit that can live on its own. A nucleus is a part of the cell, and it contains all the chromosomes. A chromosome is a long stretch of DNA. So this is a cell, and these are other parts of the cell, but we don't need to worry about those. Let's focus on the nucleus. In the nucleus, there are the chromosomes, and in the chromosomes are the DNA of the genome. So what is a gene? A gene is a part of the genome that codes for proteins. There are other genes, but we don't know what they code for, or if they code for anything at all. And a gene is also the basic unit of inheritance. Parents pass genes to their offspring, the information for traces in genes. Proteins or segments of proteins are coded by genes. There are many different jobs and functions in the body. There are around 20,000 protein coding genes in the human body. Genes are tiny parts of chromosomes, and genes determine many different things about you, like your eye color, hair color, and more. So, let's watch this. Okay. So this is a chromosome. Inside the chromosome is DNA, and a tiny part of the genome is a gene. So as you can see here, genes are tiny parts of chromosomes. So that's the chromosome, that's the gene, and it determines a trait, for example, eye color, and that's the genome. It'll probably be a little bigger than that though, and that's the DNA. So DNA is the information molecule for all living organisms. This is a cell. Inside it is the nucleus, and here are 23 pairs of chromosomes. And this is the other part of the cell. It's called a mitochondria, and that's a mitochondrial DNA. So, about 3 billion nucleotides are in the human body. A Japanese flower called Paris japonica has about 150 billion nucleotides, and it's 50 times the size of a human genome. The Human Genome Project produced the first draft of the human genome sequence, and it took 300 million worldwide to do it in 13 years. Today the cost to do it is $600, and it only takes a few days. So the biggest genome is a plant called the New Caledonian fern has the biggest known genome at 160 billion base pairs of DNA. And and the smallest known genome is a bacterium called Carcinella rudi, and it has 160,000 base pairs. So here's a really small part of the genome, and 23 pairs of chromosomes, and it's a sequence of 4 nucleotide bases, A, C, G, and T. And the total size of the genome is 3 gigabase pairs. Human genome databases provide the complete list of genes and the functional information to serve as a periodic table for biomedical research. So this is my first high-level question. How do cells make RNA and proteins from DNA? So DNA is a nucleic acid, but it's not the only one. Today we'll be learning about one other nucleic acid called RNA. So RNA stands for ribonucleic acid, and here we'll be comparing and contrasting DNA and RNA. So DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. DNA is a sequence of 4 nucleotide bases, A, C, G, and T. But RNA doesn't have the base T, which stands for thymine. A also stands for adenine, C stands for thymine, G for guanine. So instead, RNA has the base U, which stands for uracil. And since A pairs with T and C pairs with G on the DNA, they attach to each other, instead in RNA, A pairs with U and C pairs with G. So while DNA stores the genetic information, RNA converts the information to make proteins. So there are 2 types of processes needed to make a protein, and they're called transcription and translation. And this is how you make a protein. So DNA stands for deoxyribonucleic acid. It's a sequence of 4 nucleotide residues, A, C, G, and T. RNA stands for ribonucleic acid. It's a sequence of 4 nucleotide residues, A, C, G, and U. And a protein is a sequence of 20 amino acid residues. And amino acid is the basic building block of a protein. So the first process to make a protein is called transcription. So the DNA is unzipped, and a type of protein called RNA polymerase moves along the DNA and makes a long strand of RNA. This is called messenger RNA, or commonly known as mRNA. So the mRNA is notice that the mRNA is complementary to the DNA. So then the parts of the DNA that are not, the mRNA that are not needed are cut out. These are called introns, and the parts of the mRNA that survive or are not cut are called exons. So then the exons move together to form the new mRNA, and then the mRNA goes out of the nucleus and to another part of the cell called the ribosome. Inside the ribosome, there are all of these types of RNAs called tRNAs, also known as transfer RNAs, are available. And they each have an amino acid on them, which is the basic building block of a protein. And then the tRNAs are going to arrange the amino acids into a polypeptide, which is a chain of amino acids. 1 or more polypeptides make a protein. But the tRNAs don't know which amino acid to use. So that's when the mRNA comes in. The tRNAs read the mRNA, and notice how they each read the DNA, the mRNA in triplets. These are called codons. So codon table is the genetic code for translating each nucleotide triplet in mRNA into an amino acid, or a start or stop transcription and or translation signal in a protein. So there are these codons, and and if you choose one codon, it translates into one amino acid, and then one or more of these amino acids become a protein. And our ribosome is also made up of rRNA, also known as ribosomal RNA. So the tRNAs read the DNA and make a chain of amino acids, a polypeptide, and one or more of the polypeptides make the protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
So those are the introns that are being cut off.
The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein. The